Good morning, good afternoon, good evening to everybody. Welcome to the VitaFlow Symposium. My name is Tricia Rutherford and I'm Head of Research and Development at VitaFlow. It gives me absolute great pleasure today to introduce two individuals who've been collaborating with VitaFlow for the past decade or so. These two individuals are Professor Simon Heals and Professor Matthew Walker. This collaboration has been going on for approximately a decade now, and Matthew and Simon are representatives of a wider team, but they're going to introduce the project to you today. Firstly, Simon will introduce the background biochemical aspect of the project, and Matthew will outline the successful clinical trial results. This collaboration has been focused on the use of medium-chain triglycerides, a unique blend, and its effects on the dietary management of drug-resistant epilepsy. This collaboration has taken us from an idea through to laboratory work and through to a successful clinical trial, which has now been completed and results have been published. Firstly, Simon is going to present the biochemical background. Simon is the UCL Chair of Clinical Chemistry and Head of Chemical Pathology at Great Ormond Street Hospital and the Neurometabolic Unit at the National Hospital for Neurology and Neurosurgery in Queen Square in London. Matthew Walker is Professor of Neurology at the Institute of Neurology, Queen Square, and also Consultant Neurologist at the National Hospital in Queen Square. So it's my great pleasure now to hand over to you, Simon, to describe the background biochemistry of the project. Thank you.
Thank you very much, Trish, for that introduction. It's a great pleasure to talk about this project and give you a bit of a biochemical background, particularly focusing on decanoic acid, but not forgetting octanoic acid as well, because that combination I think is very important for understanding the biochemical basis behind what we're talking about and what Matthew will talk about in more detail with regards to the clinical trial. So when you start thinking about the ketogenic diet, you come up with figures like this from the literature that show an enormous amount of things that may or may not be going on. And so it can be quite daunting, but at the same time, it's good to try and unravel and see if we can work out some of the biochemical changes and also some of the biochemical key players that are in the diet that may elicit the responses in a favorable way. And one of the particular areas that we're focused on and have been focusing on for many years now is the role of the ketogenic diet and mitochondrial content and mitochondrial function and the ability to generate ATP. And we're interested in how the fatty acids of the diet, in particular, our starting point was octanoic acid and decanoic acid from the MCT diet, and how that may affect mitochondrial biogenesis. Because I like this slide, it's the mitochondria right at the center, and that reflects the area of our particular area of our research in our laboratory. So why think about the, why did we come to think about mitochondrial function and the ketogenic diet? Well, this goes back to a meeting I had attended in 2007 with Trish, who just introduced me, and her colleague Maura from VitaFlow. And I was talking about neurotransmitter disorders, but I did listen to a talk on the ketogenic diet. It seemed enormously complicated, and very hard for some patients to adhere to. And we were just talking and saying it'd be wonderful if you could find the magical component to ascertain how the diet is working. And I did tell them about a publication of mine back in 1991, where I used as a control group trying to set up a diagnostic test for a disorder called medium-chain A-sub-CoA dehydrogenase deficiency. How the patients in the control group that I was using were patients on the MCT form of the diet, and how they had very high levels of octanoic acid and decanoic acid, C8 and C10. And we started to discuss whether these fatty acids could impact on mitochondrial function in any way. And so from that tea break meeting, coffee break meeting, we, VitaFlow very kindly funded a number of laboratory-based PhD students that enabled us to answer the question, are there particularly fatty acids that can influence or impact on mitochondrial function? But why even think about mitochondrial function? Well, it's very interesting to note that 2% of our body weight is our brain, but it uses 20% of our oxygen consumption. So it's very oxygen hungry. That's because it's very, very heavily dependent upon mitochondrial content and mitochondrial function. Because energy in the form of ATP, which is the energy currency of the cell, is needed to restore electrical balance along our nerves, along our axons. So when an action potential is going along, then you get these changes of sodium, potassium that need to be restored to allow for electrical neutrality. Otherwise, if it's not restored, then this could be a contributing factor for epilepsy. So by an extrapolation, then, if you can correct or improve energy status in the form of ATP generation, or the ability to generate ATP, this may be beneficial in patients with epilepsy, because there is a growing body of evidence to show that epilepsy and seizures are associated with a loss of mitochondrial function. So correction, improvement of mitochondrial function was the hypothesis that we were pursuing, because that would increase or restore ATP levels that would allow for the restoration of the appropriate sodium potassium gradients to be achieved. So our focus of our work is the mitochondrion. I don't want to spend a lot of time talking about how they work. You can meet me later or email me later if we want to have a long discussion about this. But these components of the mitochondrial electron transport chain pick up electrons, hand them on to their neighbors using mobile electron carriers. And as they're moving these electrons, these are derived from metabolic pathways such as fatty acid oxidation, the TCA cycle, the Krebs cycle, the citric acid cycle, whatever you want to call it. These are all the classical pathways of energy metabolism. They feed in ultimately to the electron transport chain that then uses energy in a different form to move protons into this intermitochondrial membrane space to generate a proton gradient. Now, I'll mention this gradient a bit later on in my presentation, but this ability to generate the gradient is very important, because that's how you form ATP. So you need all these components of the electron transport chain to work correctly in an ordered manner to allow the our cells in the body, particularly in the brain, as I said, very energy dependent, our brains, to generate ATP, the energy currency of the cell. And so if it's lower, if its function is decreased, as has been evidenced in some of the epilepsies, then you may have an energy deficiency state, which could be a contributing factor to epilepsy. So there's a lot of focus on the mitochondrial electron transport chain and mitochondrial content with regards to a target for treating epilepsy. And as I mentioned in my earlier slide, mitochondrial biogenesis, generation of new mitochondria, has been associated with ketogenic diets. So over the years, funded by our colleagues at VitaFlow, we've been able to show that decanoic acid, but not octanoic acid, is able to elicit mitochondrial biogenesis. This is as a marker enzyme known as citrate synthase, and the complex one. So this component measured in our laboratory is increased when cells, neuronal cells, are exposed to decanoic acid, but not octanoic acid. So an increase in mitochondrial content and an increase in mitochondrial enzyme activity. But ketone bodies, up to very high concentrations, up to millimolar levels, beta-hydroxybutyrate, acetoacetate, no effect on these parameters that I've just mentioned here. You can go to electron microscopy. These are control cells, and these are cells treated with decanoic acid. And our very experienced electron microscopist said to us, you've got a lot more mitochondria here. They're a lot more electron dense. He didn't know which were controls and which were treated, but these are the decanoic acid treated. So we have biochemical markers and electron microscopy saying there's something going on with regards to mitochondrial content when the cells are exposed to decanoic acid. I mentioned to you earlier on, when I was talking about the mitochondrial electron transport chain in a very quick and simplistic way, that you need to generate a proton gradient. That's used to make the ATP. That's also known as a mitochondrial membrane potential. And what we were able to find with one of our PhD students is that when you expose cells to decanoic acid, you increase the mitochondrial membrane potential, which it can be argued improves the efficiency of those mitochondria to generate ATP. Again, decanoic acid, but not octanoic acid having a beneficial effect, or having its effect, and that is significant over the octanoic acid treated cells or the control cells which just received the vehicle only. And then we've moved on and looked at mitochondrial DNA content, content, and Tom Baldwin, who was attending this meeting, did this work, and he was able to show that the mitochondrial DNA content was also increased. Because we need to remember that that electron transport chain is made as encoded for by both nuclear DNA and by mitochondrial DNA. So you need both these genomes to work together to generate a fully assembled mitochondrial electron transport chain. And here what Tom has shown is that the decanoic acid increases the mitochondrial DNA content, which supports our observations both at the mitochondrial DNA level, and it supports the biochemical findings and the electron microscopy findings that I showed you earlier. Now, if you're going to have more and more mitochondria, you've got to remember mitochondria use oxygen and convert that to water. There's always a little bit of leakage, and they generate reactive oxygen species and molecules such as hydrogen peroxide. And so it's always good that you have very good antioxidant capacity. And what was very nice in some of our early studies, we were able to show that decanoic acid increased the content of catalase within the treated cells. And catalase can remove offending molecules such as hydrogen peroxide, and so combat any side products that may be produced as a result of having more mitochondria. So more mitochondria, a good thing on one hand, you're going to get more molecules such as hydrogen peroxide being produced. And here we are able to show that catalases increase when the cells are exposed to decanoic acid. So we were using levels of around 250 micromolar. And one of the early questions we got, well, how can that build up in the brain? Is it not just going to get metabolized away? Because these fatty acids are substrates for fatty acid beta oxidation, and they could therefore get metabolized away, and so would not, the decanoic acid would not build up. And I thought that was an excellent question, but others had shown that in the brain, decanoic acid in animals treated peripherally with decanoic acid then elevated their plasma levels, but they also elevated their brain levels to levels that we were seeing capable of causing mitochondrial biogenesis. So it's very exciting to see that it does actually build up. But how does it build up was one of our main questions that we were asking. So we looked at the metabolism of these fatty acids. We looked at decanoic acid and octanoic acid by using a label, and then seeing the amount of labeled carbon dioxide. That's the ultimate end product of the metabolism of fatty acids. So it's telling us how well they are metabolized compared to each other in these neuronal cells, and comparing that to the glucose. Now, glucose is a preferred substrate. You can see it's being very well oxidized to carbon dioxide, as we would have expected. But if you look at the octanoic acid and the decanoic acid, we saw a differential effect, which is very intriguing to us. Octanoic acid is much better at being oxidized by neuronal cells compared to decanoic acid. In fact, decanoic acid is only 20% of the oxidation rate of octanoic acid. So these cells in neuronal cells are being handled, so these fatty acids are being handled differentially by these neuronal cells, which is extremely interesting. So why was that the case? Well, we started to think about the carnitine shuttle, because the carnitine shuttle is low expression in the brain. There's a low expression of all the components of the carnitine shuttle within the brain. Now, the carnitine shuttle takes fatty acids above a certain chain length, because those longer chain fatty acids cannot get into the mitochondria for metabolism. So they need to have a new system. I see this like a passport control system. And so the fatty acid molecule is linked to a carnitine molecule, which then, this is catalyzed by the enzyme carnitine palmitoyltransferase 1. And once you've made this acyl carnitine, as it's now known, it can cross in from the cytosol through the mitochondrial membranes, translocated into the mitochondrial matrix, so this transporter, and then another enzyme does the reverse reaction, the carnitine palmitoyltransferase 2, to liberate the fatty acid that can be metabolized for fatty acid beta oxidation. So it's essential for some fatty acids, but not others, that you have a fully functional carnitine shuttle. Now remember, there's low expression of this shuttle in the brain. So if you are a fatty acid that needs the carnitine shuttle, then you might not be that lucky, and you might not be able to get into the mitochondrial matrix. But if you don't need the shuttle, you can go through unhindered and get metabolized via the beta oxidation pathway. So we did some very elegant experiments, and we used an inhibitor of the carnitine shuttle, the first part, the carnitine palmitoyltransferase 1. We blocked that. And what we found was that when you blocked this, you only inhibited octanoic acid oxidation, that's the metabolism through to CO2, by 34%, but decanoic acid was blocked by about 95%, and palmitic acid, which is even longer, 16 carbon units, was blocked by 99%. These two were very, very similar. And what these data are showing us is that the decanoic acid has a much more of a stronger requirement for the carnitine shuttle compared to octanoic acid. So remember, the brain has lower levels of expression of this system. So it's plausible then that decanoic acid can build up in the brain because it can't enter the mitochondria so well and therefore can't get metabolized, whereas octanoic acid can act as a fuel and be metabolized. So we have a mechanism, we believe, whereby decanoic acid can build up in the brain because of its reliance on the carnitine shuttle, whereas octanoic acid can go through relatively unhindered. So if you look at that and extrapolate that a little bit further, this is some work from another laboratory which supports our findings. If octanoic acid can be metabolized independent of the carnitine shuttle, and your brain receives a lot of octanoic acid, then it can be metabolized through, and not all of the fatty acid breakdown products can enter the TCA cycle. So you get builds up of molecules that form ketone bodies. That's how you generate ketones from excessive fatty acid breakdown. So if octanoic acid is dependent on the independent, I should say, of the carnitine shuttle, octanoic acid should be ketogenic. And in this study on a neural cell line, we've got octanoic acid generating ketones in the form of beta hydroxybutyrate. But look what's happening. Decanoic acid is not ketogenic. In this model system, the fatty acid is not being metabolized to generate ketones, but the octanoic acid is being metabolized to generate ketones. This fits very nicely, I'd even say beautifully, with our hypothesis that this is carnitine independent and can be metabolized through to ketones, whereas decanoic acid is metabolized independently and because of its reliance on the carnitine shuttle, and therefore can build up. So if you think about it a little bit further, a little bit might get through the decanoic acid, but if you've got octanoic acid dominating the beta oxidation pathway, it's being metabolized all the time, then it will actually protect the decanoic acid even further. So when you add octanoic acid, so you've got a decanoic acid to octanoic ratio of 80% decanoic acid, 20% octanoic acid, actually there's a sparing effect even further. So when octanoic acid is present, any decanoic acid that does sneak through into the mitochondrion, it can't get oxidized because the octanoic acid is holding up and hogging the system, if you like, to allow for the fatty acids to be, that fatty acid to be metabolized. Yet another factor then that could increase the levels of decanoic acid within the brain. The dependence on the carnitine shuttle, causing a buildup of decanoic acid, and any that does get across doesn't get metabolized very easily because octanoic acid is tying up the enzymes of beta oxidation. So this is a sparing effect on decanoic acid, even less oxidation when there's C8 octanoic acid present. What has been very exciting in all this work is we've now extended to a mouse model with colleagues in Australia, and it's just submitted for publication at the moment, but we can show that when you have an 80-20 ratio of octanoic acid to decanoic acid, there is good seizure control, a better survival, and interestingly, when you have that mixture, you get a greater buildup of decanoic acid in the brain compared to decanoic acid on its own. So this is a mixture of decanoic acid and octanoic acid, 80-20 ratio. We've got this buildup of decanoic acid greater than we have the decanoic acid on its own. That sparing effect of the octanoic acid upon the decanoic acid is showing in this animal model now within the brain. And like we saw in the cell culture, we see an increase in the mitochondrial marker. Citrate synthase has increased. In the 80-20, it's particularly increased. And another antioxidant, glutathione, is also increased in that 80-20 ratio. So it's very nice just to see our findings in the laboratory translate into this mouse model system. And then some great colleagues and collaborators, and Matthew Walker will be following me in a completely different system, which I'm sure he'll talk about, seizure control this is an acute system now, as opposed to a more chronic system I've just shown, is that decanoic acid, but not octanoic acid. There's decanoic acid here, octanoic acid here. This is a reduction in seizures in this model system. Is very, very impressive for decanoic acid when compared to octanoic acid. So a different model system, but showing these two fatty acids behave in a very, very different way. So when I try to summarize this, and I know it's only been a 20-minute presentation, I'm very happy to take questions later. Here we have decanoic acid, and here we have octanoic acid. Decanoic acid, and we've published this and shown that it can bind to PPAR gamma. That's the binding that is necessary to cause the mitochondrial biogenesis. So decanoic acid is recognized by PPAR gamma, octanoic acid is not. So this is, it binds to PPAR gamma, and this causes mitochondrial biogenesis. I've told you that decanoic acid metabolism is quite sluggish because of its reliance on the carnitine shuttle within neuronal cells. So therefore, it doesn't break down and it doesn't generate the ketone bodies via acetyl-CoA. However, octanoic acid can move through unhindered, and therefore it can be broken down by beta oxidation, builds up acetyl-CoA to form the ketone bodies. Now, if decanoic acid is stimulating mitochondrial biogenesis, increasing the enzymes of the TCA cycle, the octanoic acid component of this mixture of 80-20 is therefore a valuable fuel source, perhaps, to fuel the TCA cycle to generate energy, either directly entering the TCA cycle or via the ketone bodies. Now, I've also told you that decanoic acid in the model system has an acute effect on the AMPA receptor, and I'm sure we'll hear about more of that in the next presentation, but you need that build-up still. So that reliance on the carnitine shuttle is permitting the decanoic acid to build up, not only perhaps to stimulate PPAR gamma, but also inhibit the AMPA receptor, which will be favorable with regards to seizure outcome. And finally, octanoic acid can further stop or slow down the metabolism of decanoic acid, contributing further to the buildup of the decanoic acid, allowing for mitochondrial biogenesis, and also very much the inhibition of the AMPA receptor. And remember, in other people's studies and our own implications from our findings is decanoic acid is not really ketogenic as opposed to octanoic acid. So very different ways they are handled and very different functions, we believe, with regards to how they cause protection against seizures. So very grateful to a whole raft of people. I said at the beginning, 2 or 3 of us met for coffee back in 2007. And this is a fantastic collaborative group bringing together clinicians and scientists, and the NHS, the universities, and we're extremely grateful for the funding we received from VitaFlow. So thank you very much for your attention. Goodbye.
So it's a great pleasure to be here able to present the results from our initial clinical study. So just as a bit of background, you've heard already from Simon, but conventionally the idea has been that ketones have been the main mediator of the anti-seizure effect of the ketogenic diet. Ketogenic diet developed from the observation that starvation was an effective treatment for epilepsy back in the 1920s. And in the 1920s, the ketogenic diet was devised to try and mimic starvation. And then in the 1970s, we had the medium-chain triglyceride diet that was shown to be an effective therapy for intractable childhood epilepsy. And the medium-chain triglyceride diet tends to have about 60% octanoic acid and 40% decanoic acid, and is an effective way of inducing ketosis. So we, along with Simon, were interested to know quite how these diets work. And one of the first questions we asked was whether it's the ketones or the fats that may be mediating the anti-seizure effect. And we did this in a very reduced model, which is a slice model, where we take a hippocampal slice, we induce continuous seizure activity by either adding a chemical pentylene tetrazole or by lowering the magnesium. And then we had a look to see what the effects of the fats were, of the ketones were on the seizure-like activity. And our first observation was that decanoic acid and not octanoic acid reduced dramatically here the seizure-like activity in this reduced model. We then went on to test the ketones, and we found no effect of acetone or beta-hydroxybutyrate shown here. So the epileptiform activity remained unchanged and similar to control. And again, compared to decanoic acid, which we saw an enormous drop, And similarly, we saw the same thing in a low magnesium model of inducing seizure-like activity in these slices. So we started to investigate, along with Simon, what effects decanoic acid could be having. And along with the work that Simon's done, we've also found that decanoic acid can have effects on receptors and channels, and that maybe these acute effects on receptors and channels, along with the metabolic effects it's having, may be mediating its anti-seizure effect. But this led to this idea of trying this decanoic acid rich diet in humans as a treatment for epilepsy. And we were using K-Vita, which had a ratio of C10 to C8 of 80 to 20, as opposed to 40 to 60 in the conventional medium chain triglyceride diet. And we set up a study that was mainly there to evaluate tolerance, acceptability, and compliance. And as an added measure, we looked also at seizures. And this was a 12-week prospective study. It was a feasibility study. It was in 40 children. The aim was to have it in 40 children, 40 adults. And we gradually stepped up the dose of K-Vita in adults and children over the 12-week course. And the dietary restriction was there just to maintain calorie intake rather than to induce ketosis. And we, of course, wanted to see whether we had significant ketones. We wanted to see what happened to decanoic acid, weight, side effects, and seizures. And just overall, just to take an example of what happened to the carbohydrate, protein, and fat compared to the medium chain triglyceride and classic ketogenic diets and normal diets, you can see here with K-Vita, we increase the amount of fat in the diet because we're giving a fat-rich supplement. But there was far less restriction of carbohydrates, you can see, compared to the MCT and the classic ketogenic diets. And protein remained about stable. We initially wanted to look at two different groups, and those were the groups that we thought this sort of diet would mainly be used in. So in children, we picked people with a epilepsy syndrome or diagnosis of genetic epilepsy, and these children had severe epilepsies or other paroxysmal events. So we also had people with alternating hemiplegia of childhood, and also people with GLUT1 deficiency syndrome who would have a movement disorder in addition to seizures. And you can see here the majority were genetic epilepsies, including Dravet syndrome. In adults, we sort of picked all comers, but just people who are very resistant and people whom we would consider referring to our ketogenic diet service. And overall, we had about 40 children and 40 adults initially consented. Unfortunately, about 16 dropped out, and the main reasons for that were an abnormal acetylcarnitine profile at baseline, in particular in adults, and then a host of other reasons of people withdrawing for actually starting K-Vita. In the end, we had just over 60 people, 35 children, 26 adults who started K-Vita. And when we look at this particular population, they were a very refractory population. They all had frequent seizures. The median number of anti-seizure medications tried in children was 3, and in adults it was 10. So they tried 10 anti-seizure medications and still continued to have seizures. And importantly, about a third had previously tried the ketogenic diet, but had to withdraw because of personal reasons or adverse effects from the diet, or the diet being ineffective. Overall, by the time the study finished, which was after 12 weeks, we had about 2/3 of people completing. So about 2/3 of children and 2/3 of adults. And this compares very favorably to studies of medium chain triglyceride diet or the ketogenic diet. And overall, the diet was well tolerated. The main reasons for withdrawal were gastrointestinal side effects. And we found that often these occurred early on, probably because at the beginning we were titrating the dietary supplement up too quickly. And in fact, later in the study, we titrated more slowly and people tolerated the diet better. And then in the odd patient, there was worsening of seizures or behavioral change for reduced appetite. The main side effects overall were, again, mainly gastrointestinal. They improved with time, so very often people would complain of them as the diet was titrated up, and then over a few weeks these side effects resolved. There were rare occasions of mood swings, behavioral change, decreased appetite and fatigue, but overall these were mainly gastrointestinal side effects. Importantly, over the 12 weeks, there was no significant change in body weight in either children or adults. Now, one of the first questions is, do we actually induce ketosis with this diet? And so here we have a measure of beta-hydroxybutyrate. And if you take about 1 millimolar as being evidence of ketosis, we only had 3 children in the final visit and 1 adult who actually would count as ketotic. And even then they had low beta-hydroxybutyrate levels. In contrast, we see a marked increase in octanoic acid by the 3rd visit in both adults and children, and of course decanoic acid, which was the fat that we were trying to increase in the plasma. And you can see here that the decanoic acid concentration was somewhere around about 100 to 200 micromolar. So what about efficacy? So we found that the diet was well tolerated. We found that in the majority of children and adults, it did not induce ketosis. We found that decanoic acid levels went up. We had efficacy data in about 78% of patients. We were unable to retrieve efficacy data in the others. And we looked at the change in seizures from the baseline visit, visit A, through to the visit in the middle of the study, 5 weeks, and the visit at the end at 12 weeks. And we found a significant reduction. This is log seizures, just to show you the number of seizures that these patients are having, log seizures per week. So they're having vast numbers of seizures. And you can see that there's a significant decrease both by the 2nd visit and by the 3rd visit in the number of seizures that patients were having. So that a significant number of people were having their seizures reduced and some even having their seizures stopped. We looked to see whether the reduction in seizures correlated with the beta-hydroxybutyrate concentrations, negatively correlated with those, or whether it negatively correlated with C10. And we found no correlation with beta-hydroxybutyrate concentrations, but we did find that there was a correlation with C10 levels, indicating that the C10 was probably mediating this seizure reduction. And what about opinion of it? Well, overall, all the parents said that beta shot was easy for the child to take. They either agreed or strongly agreed with that. And 86% found that it was very easy to follow the advice. So the advice was really about reducing sugar intake rather than a strict diet. In adults, similar number found it was easy to use and that it was easy to follow the advice about reducing sugar, indicating that you need minimal dietetic input in order to maintain this diet. So in conclusion, preclinical evidence indicates that decanoic acid, a medium-chain fatty acid, has anti-seizure effects, that K-Vita, the medium-chain triglyceride food with a high C10 to C8 ratio, is well tolerated and easy to use. The side effects are mainly gastrointestinal. Few people developed significant ketosis. And in this particularly refractory group, we saw a median reduction of 50% in the number of seizures, with some people reacting particularly well to the diet. In particular, we saw this in people with GLUT1 deficiency. The efficacy correlates with C10 but not with beta hydroxybutyrate levels, indicating that this is probably, or supporting the fact that this is probably the C10 that is mediating the effect, and not any change in the ketones. With that, I'd like to thank all the people involved in this study, in particular Natasha Scholler and Michael Orford, and the senior investigators in the study, which are myself, Helen Cross, and Simon Hills. And thank you very much indeed for your attention.